So today I want to talk to you a little bit about answering some questions. So I want to talk about what is the analysis of computed tomography I'm using, why I'm using polymeric self-assemble and gold nanoparticles for computed tomography analysis, how does it work? It's the objective of my research, and does it work, which is the most important question of all. So, computer tomography is one very commonly used imaging technique using clinical applications. So, this is a very famous picture that everybody knows. Computer tomography is based on X-ray attenuation coefficients that pass through the body, and different materials give different attenuations depending on the density of the materials. So this technique have many advantages such as short image times, high resolution images, and good tissue penetration. Unfortunately, there are some bad disadvantages as exposure to ionizing radiation, difficulty of differentiation between soft tissues, since the water and oxygen are not very high in atomic number. And one of the main characteristics that I'm exploring in my research is the linearly relation to gold concentration, or like the contrast agent concentration. As you can see in the image of the phantom, you have the most concentration of the approximately 20 milligrams per mil of gold, opposed to the 2 or the 5 milligrams per mil. So the objective is to have enough gold concentration that I can get contrast using computed tomography. And why I'm using polymeric self-assembly as to answer this question, and to explain what is polymeric self-assembly a little bit. So polymeric self-assembly is basically composed by a hydrophobic block and a hydrophilic block that forms different structures in water based on those two components. So you can have very simple structures such as nanofibers to very complex structures to polymerosomes, dendrimers, and the one that I'm interested in is the micelle. So why use polymeric self-assembly? So you can encapsulate hydrophobic substances due to hydrophobic block. You can manipulate the physical and chemical properties of the polymer self-assembly through different block sizes and chemical properties of the block polymers, and created a protection environment that can increase blood circulation in the body. And you can change also chemically to change your structure to have a response via passive or active targeting on your cellular uptake. And why the gold nanoparticles and how they fit into this is the gold nanoparticles have been widely studied for different diseases, treatments, and diagnosis, and this is mainly to their synthetic versatility that enables different sizes, shapes, and properties of surfaces. For example, their easy attachment to amines and thiols can allow gold to bind with different molecules for different treatments, such as gene therapy, protein, and drug. They do not present acute toxicity based on recent research. And the main characteristic that I'm interested in is their high X-ray absorption coefficient, which can allow you to do the X-ray CT imaging and the radiation therapy. And to answer the how part of this presentation is how do these particles are functionalized. So the gold nanoparticles are first reduced by using sodium boron hydroxide, and then they are functionalized using a hydrophobic polymer ended thiol that attaches to the surface of the nanoparticles. The polymer I'm using is a polycaprolactone ended thiol, and the size of the nanoparticles are close to 8 to 10 nanometers in the core. And how the self-assembly works after the functionalized gold nanoparticles with PCl thiol, they are assembled with the block of polymer composed of PEO PCl. So it's the same hydrophobic block I'm using to functionalize gold nanoparticles. And the hydrophilic block is composed by polyethylene oxide, and the hydrophobic block is PCl. So you have the nanoprecipitation through, so you have the mixing of the particles with the block polymer in a organic solvent, and then you load them into the syringe to have the nanoprecipitation, where they are going to form the gold-loaded polymeric micelle. And does it work? So the first thing that we analyze is size, which is one of the most characteristics that are going to influence the cellular uptake and blood circulation. So because you have different parameters, you can have different sizes, distributions, and average. So in here, I'm showing you different things that can influence those particular things using DLS. So first of all, you can change the amount of water you can use. So you have a different water content by the end of the mixing and the nanoprecipitation. So in here, the first on your left, you can have different size averages, such as 150 if you use 1 ml of that mix into 2 ml of water. So 1/3 of the mix is polymer with gold. Or you can have a more dilute sample that creates a bigger particle. And you can also change the gold to copolymer ratio, and that gives smaller sizes. So I'm using more gold than copolymer in this experiment. So you can have 20 times or 50 times more, 100 times, or you can even have like an inversion of the ratio. And then you have different sizes as well. So the parameters that can affect size and stability of the particles are the gold concentration, the size of the polymeric block, because you can have also different sizes of PCl and PEO, and that would definitely influence the sizes and also the stability in the gold loading. And you can have different ratios, and you can also have the different amount of water. But this works. So I was able to prove by using TEM that the particles are actually encapsulating into the polymeric micelles. So in here you have the TEM staining of the particles. So you can see that the clear particles inside are spherical, and you have the black dots represented by the golden particles. So in here you have the two different ratios of the 50 times more golden polymer and the 20 times golden polymer. So the sizes are the average size are influenced by how much gold is encapsulated in the particles. And lastly, so does it work with computed tomography? So as far as we're doing the experiment, these are the functionalized gold nanoparticles. They have a concentration of approximately 3 to 4 milligram per mil. So it actually see contrast. But unfortunately, since they are hydrophobic, they are in organic solvent. So the main thing to do is after encapsulation, you can have enough concentration so you can get contrast. So this is where it gets tricky, and this is why I'm changing so many variables so I can see contrast using the CT experiment. So you see that there's also a difference in contrast according to the gold concentration. So if it is a lower gold concentration than the image noise, you don't see anything. And the more gold you have, the more unstable the particles are as well. So you can see that there is a lot of precipitation, and that's the tricky part, and that's what I'm studying. So first, I wanted to thank you, my supervisor and my co-supervisor in Devon, which are the people responsible for the CT imaging part of my research, and they're being very helpful. And to summarize, I wanted to say that the gold nanoparticles were successfully functionalized and encapsulated, and they have demonstrated to have contrast under CT, but unfortunately, only the functionalized ones in organic solvent presented contrast. But futurely, different cellular uptakes and different parameters are going to be changed to change CT contrast and cellular uptake, as well as radiosensitization assays will be performed to assess the treatment efficiency with those particles. So I would like to thank you and answer any questions you might have.
Great. Thank you very much, Talita. So I'm pleased to open the floor to questions from the audience. Please either submit a written question or raise your hands. And to give everyone, oh, actually, I do have a question already. Thank you for typing so quickly, Marcel. So our first question is from Marcel Bally, one of our research leaders. And he asks, I don't understand how you plan on utilizing these gold nanoparticle / polymer particles. Is it really just contrast enhancement? Do you see a therapeutic use?
Yes, so because they are encapsulated into polymeric micelles, so the first goal was to obtain contrast. And so after that, we're going to do some chromogenic assays to treat them with using radiation, so using X-ray absorption. And also, if time allows, we're going to co-encapsulate chemotherapeutic agents with those gold nanoparticles and see how they interact with the cells.
Great. Thank you, Talita, for your answer, and Marcel for your question. And so while we see if anyone else has any questions, I have a question for you, Talita. Could you speak to the gold nanoparticle functionalization, how you figure that out?
Yeah, actually, I'm going to share my screen again.
Of course.
Just to show a little bit of the analysis that I do. So after gold nanoparticles functionalization, I do thermogrammetric analysis to assess functionalization, because I do have a process of functionalization that removes the unattached chains. So I check by TGA if the polymeric chains are removed from the system. And using this, you can see the broadening, a little bit of shift of the peak when the particles are functionalized and after that encapsulated.
Great, thank you. And our next question is from Ariadne again. Thank you very much, Ariadne. So she says, hello, Talita, good talk. You said the only one that works was in an organic solvent. Do you need to, can you change the solvent to water or a more biological compatible solvent for the next studies?
Yeah, so the particles are inorganic solvent because they're just functionalized with hydrophobic polymer. So since PCl is hydrophobic, I can change to water, otherwise we're just going to aggregate and precipitate. So that's why I'm trying to encapsulate it. So there are not many studies doing hydrophobic gold because of that issue, because it's a very hard thing to do, especially in the concentration that you need. So unfortunately, they are an organic solvent and I can just use that. I need to encapsulate in water and then remove the organic solvent by dialysis.
Great, thank you. And so I'll ask, I don't see any other questions, so I'll ask a final question. I was curious, could you speak to how the radiosensitization by gold nanoparticles work?
Yeah, I do have a very messy slide to explain a little bit. It's just like actually a cascade of events. So that relates a little bit with Marcel's question before. So what gold does during radiosensitization is just creates a cascade of events. So because it's a very high atomic number, the interaction with the photon releases the electrons with photoelectric and Compton effects and a lot of those effects. And it actually creates radicals and reactive oxygen species that actually lead to cell kill. So the cells ended up killing themselves because they can't grow and do it like properly.
Okay.